Welcome back to Cell and Molecular Biology course. In this session, we will discuss the DNA versus RNA, the two key nucleic acids at the heart of all life forms. So today we will explore how they are built, how they differ, and how both are essential to life. So let's dive into this fascinating world of genetic information. Both DNA and RNA are types of nucleic acids made up of repeating building blocks called nucleotides. They are responsible for storing and transmitting the genetic instructions that determine how every organism grows, develops, and functions. While they share similarities, they also have distinct roles in the cells. So let's begin with the structure of DNA. DNA, or famous for its double helix structure, like a twisted ladder. The sides of this ladder are made up of sugar and phosphate molecules, while the steps are pairs of nitrogen bases. These bases pair in a specific way, that is, adenine pairs with thymine and guanine pairs with cytosine. DNA sugar is deoxyribose, which makes it more stable and ideal for long-term storage of genetic information. So this is the structure of DNA. In contrast, RNA or ribonucleic acid is usually single-stranded and its sugar is ribose, which contains one more oxygen atom than the deoxyribose. RNA also uses uracil instead of thymine, so its bases are adenine, uracil, thymine, and cytosine. So this difference makes RNA more reactive and less stable, but perfect for its temporary role in the cell. So this is the structure of RNA. Apart from this, RNA comes in several types, each with a specific function. Messenger RNA or mRNA carries the genetic code from the DNA to the ribosome, where proteins are made, and then the transfer where the proteins are made, and transfer RNA or tRNA, which brings the correct amino acids to the ribosome during the protein synthesis. And then the ribosomal RNA or rRNA helps form the structure of ribosomes and also catalyzes the assembly of proteins. So together they form the machinery of gene expression. So about RNA types we have discussed clearly in the previous video. So now let's see the key differences in the structure, which is the sugar component. DNA is double-stranded and forms a helix, while RNA is typically single-stranded. DNA's sugar is deoxyribose, while RNA has ribose. So in the illustration, you can see the ribose sugar structure, and then the deoxyribose sugar structure, where you can see an oxygen atom is missing from the structure. So in ribose sugar, you can see an extra oxygen atom. So this is the basic difference between the sugar component. And in terms of bases, that is nitrogenous bases, DNA uses thymine, while RNA substitutes the thymine with uracil. These structural differences affect their stability and function in the cell. So you can see that purines, that is nitrogenous bases, or of two types, purines and pyrimidines. Purines are two-cycle compounds, which use the adenine and guanine, and then the pyrimidines are single-cycle compounds, which is uracil, thymine, and cytosine. So now let's see the key differences in the functions. Functionally, DNA is the master blueprint. It stays protected inside the nucleus and it stores the instructions needed for life. RNA, however, is the active player. It takes DNA's information and helps convert it into proteins, the molecules that perform most cellular tasks. So you can think of DNA as a library and RNA as the courier and translator. So the genetic information is stored in DNA, but for gene expression, that is the information, whatever information stored for it to convert into uh, an active form, about how DNA is transcribed into RNA, and then about how the RNA is translated into proteins. We have seen clearly in the central dogma of molecular biology. So this is how genetic information from the DNA is transferred to proteins by the RNA. So both play key roles in the functions of the cell. Now let's see their location. Now let's see their location in cells. DNA is mostly found in the nucleus in eukaryotic cells, while RNA is more mobile. It is synthesized in the nucleus but then moves out into the cytoplasm where it carries out its functions. But in prokaryotes, which lack a nucleus, both DNA and RNA are found in the cytoplasm, but their roles remain distinct. Coming to the stability and lifespan, DNA is designed to be stable and long-lasting, preserving the genetic information over an organism's lifetime and even across generations. RNA, on the other hand, is meant to be temporary and its molecules are short-lived, degraded after their job is done, which ensures tight control over protein production. So their relation is best captured by the central dogma of molecular biology. DNA makes RNA and RNA makes proteins. First, DNA is transcribed into RNA and then RNA is translated into proteins. So without this flow of information, cells wouldn't function and life as we know it would not be possible. So both DNA and RNA are essential in their own right. DNA ensures heredity, passing genetic information from parent to offspring, and RNA ensures function, translating those genetic codes into action in the form of proteins that build and maintain the cell. So there are so they are two sides of the same coin, each vital to life. DNA is DNA is the stable, long-term blueprint stored in the nucleus, while RNA is the messenger and worker moving between the nucleus and the cytoplasm to turn DNA's instructions into real cellular functions. So their partnership underpins every process in biology. Thank you for joining me in exploring the world of DNA and RNA. I hope this comparison helped clarify their similarities and their differences and their essential roles in the life of a cell. Thank you.